All right, well, good afternoon, everyone. Thanks for joining today for the fourth core seminar series for spring 22. As you notice, we're recording, so please be mindful and mute your microphone or pause your camera if you're not actively engaged in the conversation. My name is Andrew Bernard. I'm the Director of Centralized Cores at UMass Amherst. We're holding these seminars with the idea to provide awareness for the facility's capabilities and also some good work being done. To give you a chance to interact with the core staff and also folks who have been using the cores to find out ways they may help your research. Today, we'll be hearing from the flow cytometry core and a particular application from the Rotello group. As I mentioned, the seminar is being recorded, so those who can't join us today are able to watch later. All of our past seminars are online for the past 3 semesters. I'll put a link in the chat in case you haven't seen those yet. And we're always interested to hear what you'd like to hear from us. We are planned throughout the end of the spring semester, but for future events, we'd love to hear what you want to see. I'll be in the chat and I'll be monitoring messages. So if you have questions, please feel free to put them in the chat there. We'll go through the presentation and take Q&A at the end. So if you are comfortable sharing and talking as well, feel free to raise your hand at the end and I'll make sure we moderate and get you up there. So with that, we'll get to the good stuff and I'll turn it over to Amy.
Hello, everybody. I hope you had a very nice long weekend. I'm Amy Burnside, the director of the Flow Cytometry Core Facility. The Flow Cytometry Core Facility has capabilities to help you with your research. Everything from, we consult and assist with everything from experimental design to sample prep to data analysis, acquisition. And today we have David Luther from the Rotello lab. David is a 5th year graduate student, PhD candidate from SUNY Birmingham originally. And he works with, he focuses on green nanoparticle synthesis using natural products chemistry. He has several publications and is a trainee from the CBI program here at UMass Amherst. David's done a lot of work in the flow core facility with our imaging flow cytometer, the Amnis IDEAS. The image stream has 3 lasers, a blue, a purple, or 405, and a 488, and then a red laser, 640. So it's capable of 12-channel acquisition and analysis. David said to me the other day that once he used the image stream, he won't use anything else. So I asked him to present his research and tell us how well the Amnis has worked for him. David, I know it's a busy time of year, and I really appreciate you taking the time to put this presentation together for us today. And I'm looking forward to seeing your research.
Great, thanks, Amy. And thanks everyone for being here. I am going to share my screen now. All right. Yeah, so today I'm going to be talking a little bit about my research into the cytosolic delivery of proteins. And specifically, I'm going to be focusing on how we use the imaging flow cytometry techniques at the flow core to develop a high throughput method to screen different delivery vehicle candidates and achieve efficient cytosolic delivery of proteins. And so today I'll talk a little bit about the background of protein therapeutics. I'll introduce the delivery vehicles that we use on our end for protein delivery. And then I'll get into some of the nitty-gritty of how we actually can analyze the data from the flow cytometer and get really good quantitative data using these large data sets that we generate from the Amnis. And so before we get into that, first, I just want to talk a little bit about protein therapeutics. And so in the past couple of decades, protein therapeutics have really come to the limelight in a number of different ways as new treatment modalities for disease. And a couple of applications you might be familiar with: antibody drug conjugates use the specificity of antibodies to deliver small molecule therapeutics that are conjugated to the protein to their site of action to get more effective therapeutics. And especially in the past couple of years, monoclonal antibody therapy has become a real hot topic with the rise of COVID-19 and for its ability to activate the immune system against specific antigens using man-made antibodies. And so to date, there's over 130 FDA-approved protein therapeutics on the market. And what's really interesting is that all of these therapeutics are focused on extracellular targets on the surface of the cell. And what that leaves is a large area of opportunity for intracellular delivery of proteins. And so being able to get protein therapeutics or any sort of proteins into the cell, into the cytosol, opens us up to all sorts of possibilities. We can replace dysfunctional proteins or supplement proteins that have low copy numbers within the cell. We can modulate cell signaling pathways through upregulation or inhibition to control cellular phenotypes. And we can, even with new technologies for gene editing, we can selectively interrogate the genome, making targeted edits to genes and see how these edits affect the physiology of the cell and of its progeny cells. And plus, the ability to access all of these different processes in the cell allows us to visualize these processes in real time using fluorescence techniques. So all in all, being able to deliver proteins into the cell is a really, really powerful area. And part of the reason that it hasn't been explored more is because cytosolic access is really difficult, especially for large, highly charged species like exogenous proteins. And cellular uptake is actually not that hard because cells uptake materials all the time through general pathways like phagocytosis or pinocytosis, or through more specific forms of uptake like specific types of endocytosis. And generally, when cells uptake materials, they uptake into vesicles called endosomes. And these endosomes sort of work as sorting compartments for the cell to decide what to do with the material next. And 9 times out of 10, that means the endocytosed material will either be exocytosed back out of the cell, or it'll enter the endolysosomal pathway and be degraded. And if you're trying to deliver a therapeutic cargo like protein or nucleic acid, that's really bad. That's not what you want. So what researchers have done is develop nanocarriers, which can associate with protein cargo, promote uptake into the cell, and then trigger endosomal rupture so that you actually get escape out into the cytosol of your cargo. And the challenge with that is that even with the most efficient endosomal escape vehicles, recent studies have shown that you get about less than 10% into the cytosol. So generally speaking, endosomal uptake and release is an inefficient way to get things into the cytosol. And alternatively, delivery vehicles can be engineered to fuse with the cell membrane and deliver directly into the cytosol. And this includes vehicles like liposomes, which have similar chemical composition to the membrane itself, as well as guanidinium-rich carriers, which have been shown in a number of studies to passively translocate across the membrane. And the trick with all of this is we have to be able to distinguish between endosomal entrapment of our cargo and cytosolic access of our delivery cargo, because there's such a big difference. And so how we do that, one really good way to go about that is by using a good model protein cargo. And so in our lab, we focus a lot on GFP, the canonical green fluorescent protein. And GFP has a lot of advantages as a model protein cargo. It has robust fluorescence that's based on its structure. It has a high quantum yield and a relatively small size. And that small size allows it to passively diffuse into the nucleus after it has access to the cytosol. And what that means is that nuclear fluorescence is a telltale sign of cytosolic access when you're using GFP, which is really handy. And these 2 micrographs at the bottom right, you can see here, the one on the left with all those punctate little spots, that's indicative of endocytosis or some sort of vesicular uptake or entrapment on the surface of the cell. While on the right, you have that nice diffuse green fluorescence, as well as diffuse nuclear fluorescence that indicates that the protein is able to access the cytosol and diffuse throughout the cell. So in our lab, we're in the chemistry department. So we look at things from the nanocarrier perspective. And we've developed a few different carrier vehicles over the years to deliver proteins into the cytosol. And one of the most efficient and versatile vehicles that we've studied and that we still work with are these polyoxynorbornene imide polymers with guanidinium functionalities. So we call them PONI-Guan for short. And these PONI-Guan polymers we use to self-assemble with our protein cargo. And the way we do this is by modifying our protein cargo to feature a polyglutamate tag and terminus. And the polyglutamate tag can interact with the guanidinium functionality through guanidinium carboxylate interaction to form these supramolecular nanocomposite structures. And we've seen through characterization that these nanocomposites are discrete and relatively monodisperse in size. They have, they feature cationic surface charge, which is important for interaction with the anionic cell membrane. And what's nice is that we actually have a pretty good range of formulation space to play with these nanocomposites. And the way we do that is through formulation by charge. And when we do that with these, we use the ratio of guanidinium on the polymer to glutamate on the protein to give a sort of G to E or G positive to negative ratio, which is sort of analogous to an N to P ratio when you're doing plasmid delivery. And we can see that when we incubate these guys with cells, we get that diffuse cytosolic fluorescence that we're looking for. In these micrographs over on the right, you can see delivery of GFP into HEC293 T cells. And what we're doing here is using 4 different molecular weight homopolymers, ranging from about 8K to about 60K in size. And you can see that there's a clear difference in efficiency in number of cells that experience cytosolic delivery between 8K and 60K with that molecular weight. And so with a family of homopolymers like this, between that and our formulation by charge ratio, we have a lot of formulation space to play with when we want to optimize delivery conditions. And it's, frankly, it's too many permutations to go through to analyze just by microscopy. I do have an asterisk there because I should say that the light microscopy facility at UMass really is top notch. Jim's got some fantastic instruments and an analytical software that can allow you to do semi-high throughput measurements and imaging. But when you're doing microscopy, there's inherently limitations to quantification. So we turn to flow cytometry. And flow cytometry is designed to give quantitative data. And using traditional flow cytometry, we can get intensity measurements for our cell populations. And you can see at the bottom left here, we have mean fluorescence intensity for different G to E ratios of different polymers. And the efficiency by MFI sort of lines up with the micrographs on the right, which is good. But you can also see that there's sort of, some of the trends are a little bit wonky with MFI. And the reason for this, in part at least, is because traditional flow cytometry can't distinguish between those punctate spots and that diffuse cytosolic fluorescence. It just reads fluorescence. So, how do we go about overcoming this? We turn to imaging flow cytometry. So this is the Amnis ImageStream Mark II, the imaging flow cytometer. And looking at this, you probably say right away, that's a big black box. And it kind of is, but really it is just a flow cytometer that combines the features of flow cytometry with the morphological details of microscopy. So basically, the big difference when you're using an imaging flow is it works exactly the same as traditional flow cytometry, same information that we will get from the Fortessa. Just the difference is that this will take an image for every single cell that goes through the fluidics. So what you end up with are these huge data sets or large data sets of images. Sort of similar to what you see at the bottom left there, where we have, that's an image of one cell, bright field, and then channel 7 there, which is showing the nuclear stain. And you'll get that for every cell that passes through the instrument. And what's nice is that you can get all sorts of information with this. You can do multi-magnification imaging between 20x, 40x, and 60x imaging. And depending on the size of your cell or whatever you're looking at, this could be really important. The instrument has 3 lasers, which was recently upgraded, and it has capability for 6, I believe. But really, with 3 lasers, it's 12-channel imaging. You can get pretty much any fluorophore you need. Recently, Amy upgraded this auto sampler in here, which you see at the bottom right. You just put all of your samples into a 96 well plate, insert it into that tray. You can set up your protocol for an acquisition, and then press the button, and it'll text you when it's done. So you can do this in a very high throughput way. And so this is a really powerful technology for getting quantitative data, which gives both quantitation on the flow cytometry end and also morphological details for subcellular localization of species in the cell. And so I want to look really quickly at an example of how Luminex actually gives to do this, where we are actually looking at subcellular localization of NF-kappa-B and translocation of NF-kappa-B into the nucleus after stimulation with lipopolysaccharide. And so LPS is a membrane component of bacteria. It basically is a signal to the cell that it's undergoing some sort of stress because it can recognize bacteria. NF-kappa-B translocates into the nucleus as an important transcriptional step for activating a whole bunch of signaling pathways. So these gallery images on the bottom left here, you see, this is pretty much what you get, the data that you get from the Amnis. This is what it looks like. You get these big galleries of images with all your different channels that you are interested in. And here we've got the bright field, 7AAD, which is our nuclear stain, and then our NF-kappa-B FITC channel. And you can see on the left, this is the control, where there's no stimulation with LPS, and NF-kappa-B does not translocate into the nucleus. And you can see that the cytosol is nice and evenly stained, but the nucleus is actually dark. And then on the right, where we have LPS stimulation, we get that co-localization between the NF-kappa-B and the 7AAD signal. And what they did here was they actually used one of the software wizards on the Amnis software to calculate the similarity between those two channels. And that's what this shift here is between the yellow and the blue on the right, showing that you do have higher similarity with your LPS stimulated cells, and that indicates that you have translocation into the nucleus. And these software wizards are really powerful. The Amnis has all sorts of different wizards you can use to track internalization, co-localization, all sorts of different subcellular features that you might be interested in looking at. And that's the IDEAS software, is the name of the software that the Amnis is connected to. And so this is how, this is an example of how Luminex has done it. And now we can take a look at our raw data using PONI-Guan to deliver GFP into the cell. And so here, we're going to go kind of sequentially. Again, you've got these gallery images on the left, which are pretty much the raw data that we get out of the instrument. And it's pretty nice. You have every single cell that you collect the data for. I should say that I turned up the lookup tables a little bit, probably a little more than I should have, to see, just for everyone to visualize the GFP signal. But there's a clear difference between like number 22 there at the top, where you have that single spot. That kind of indicates that you have a nanocomposite on the surface of the cell, or possibly in a vesicle inside the cell, as opposed to number 38, 2 down, where you have just diffuse green fluorescence throughout the cell, and you have overlap with the nuclear stain. And this image gallery, like I said, images every event that the flow cytometer sees. You can, I normally gate it so that it only collects things above a certain size, which you can only do by imaging flow. And so that's how you end up with your raw data, your image gallery. After you have this, second step is the same as you would do with the Fortessa. You want to isolate the single and the focused cells. That's why I've got number 2 there. After that, when we're doing this to look at co-localization between the nuclear stain and GFP, the first thing that we do is one of the software wizards. We actually use the internalization wizard. And at this point, we start to use these masking techniques. And this is the premise of how the software actually works, is what you can see at the top right, those images with the masking. That is basically applying a binary onto the image that you generate, that you collect. And then that binary is how the software actually processes the image and how you do your analysis. And so the software comes with all sorts of built-in binaries that your internalization erode is actually one of the masks. And you can also define your own mask if you decide you don't like it for whatever reason and you want to define a different one. And so we use the wizard to quantify internalization, or to gate the population that has internalized GFP. And then finally, we look at the co-localization between the GFP signal and the nuclear stain. And that's how we get our population of cells that have GFP in the nucleus that have experienced cytosolic delivery. And now we take this back and look at how this translates into our data. We can see big difference between cytosolic delivery images versus endocytosis, or a cell that's not delivered at all. And the data at the bottom right there is the quantified cytosolic delivery data. And while the general trend between the MFI data and this quantified Amnis data is pretty similar, but you can see there's definitely a difference in terms of G to E ratio. And now we can see the GD of 5 and 10 give us the most cytosolic delivery, while below that, it's not in the cytosol. So maybe trapped on the surface or in an endosome, and it trails off up higher. So this is something that we wouldn't have been able to see with MFI alone. And this shows how we can use the Amnis to get this subcellular localization data to improve the quality of our data and learn more about our systems in sort of a high throughput way. We can image thousands of cells in minutes. You can quantify in minutes. Especially with the auto sampler, you can have like hundreds of samples easily processed. And that's how we were able to use this technique to quantify our data for cytosolic delivery. And so moving forward with our systems, right now we're interested in how polymer architecture affects uptake a lot, because again, we're chemists, and there's a lot that we can do with our PONI-Guan polymers to modulate how how cells see the polymers, and how they interact with our protein cargo. And I also have how delivery can be more accessible. And what I mean by this is, in our current system, we use mutagenesis methods to actually generate that E-tag protein. And mutagenesis can be sort of a cumbersome, expensive technique, and not all labs can use this. Also, mutagenesis can affect the inherent properties of the protein depending on the protein cargo. So we're exploring more options for delivery of native proteins or chemical conjugation methods that could overcome some of these limitations. And so just in conclusion, in our lab, we explore different delivery vehicle technologies to get protein into the cytosol. Using GFP, we have a really clear readout for how the protein is able to enter the cytosol and access the nucleus. And using imaging flow cytometry, we can take a look at subcellular localization of our delivered protein cargo to distinguish basically efficient delivery from inefficient endosomal entrapment. And the imaging flow is a really powerful technique which can do all sorts of different things. And I am only scratching the surface of what we can use it for here. And I only hope, because I'm planning on graduating at some point soon, I just hope that some intrepid grad student from our lab is able to take it on to new heights and use these technologies to discover some really, really cool stuff. And with that, I just need to acknowledge Amy. Thank you so much for having me here today. CBI program, Department of Chemistry, my committee members, and of course, the Rotello lab. Thanks so many of you guys for being here. And with that, if anyone has any questions, I am happy to take them.
Thank you, David. Folks, feel free to raise your hands if you have a question, or just unmute, or if you want to put it in the chat, we can read it for you.
No pressure.
Okay, Joe, you have a question?
Well, I'll ask one. Just curiosity, David. The co-localization algorithms that you use, did you use the wizard to set that, or did you have to adjust it much to get that algorithm to work?
So there's a lot of different ways you can actually approach that nuclear co-localization. And the way that I did it was using two different gates, basically. I started by looking at just internalized GFP, just any GFP that was inside the cell. And that was one of the wizards that comes with the software. And then after that, because I was specifically looking at nuclear co-localization, I looked at the feature similarities between like the two channels. But as I was showing in that NFKB example, you can use another wizard on top of that to just look at similarity between the two channels. And you can do most things without modifying the protocols too much, if that makes sense.
Mm-hmm. Thank you. We've used the wizard a little bit, and it's fabulous at all the different parameters you can go through with it. So, and relatively easily. So, like it too.
Any other questions? Oh, go ahead, Amy.
I'll ask a question. So David, you were working with somebody else before you started working on the Amnis. How'd you find your transition to using the Amnis versus using microscopy or using the other flow cytometers? Was it a hard transition?
Yes. Well, so I've used microscopy a lot. I've spent a lot of time up in the light microscopy facility. The Fortessa I actually didn't learn until after I had used the Amnis. So I learned the Amnis first. And I think it's easier. I would rather use the Amnis than I would use the Fortessa, especially at this point, because I'm more comfortable. But honestly, the sample prep is very straightforward. It's just, I fix my cells, do whatever stain I need, and then it's just the volume is different, I think, with the Fortessa. But yeah, the transition between the two, I think, is pretty straightforward. It's just a software difference.
And did you find it difficult to learn the software or to understand what it meant by similarity index or anything like that? Or
I definitely had to play with the software a little bit, but the more I trust it, the better it works.
Amy, I'll ask of you, how do you handle training for the Amnis and folks who maybe haven't used it yet? Do you have a standard process you use to get folks familiar with it?
Yes, generally when they come to me with an experiment, I'll often suggest, I do have another user on here who, if we're having trouble with traditional flow, sometimes throwing in the images can help understand the biology a little better. So it's best learned in context with the experiment. And I'll sit down and help people collect their images and then let them play with the data. But Amnis is also really good if you contact them, or they have been in the past, about helping you go through some of your data and using the feature finders to find. But most people, it's nuclear co-localization. The wizards are pretty straightforward. And learning compensation a little bit can be a challenge, but I walked people through learning compensation. And so, but once they get the basics, they kind of run with it. So I just get them up and running, and usually they can go from there.
And I guess a somewhat related question, how is capacity right now? If folks are interested in using this, is there bandwidth for them to get on it?
Oh, yes, there's quite a bit of bandwidth. And I think, it's intimidating at first with all the images, but if you've done imaging with microscopy, I think people are more comfortable coming from the flow, and it's, but just using it a few times, it can get up pretty fast. But I think it's intimidating because understanding the terminology is different, like masks and features and stuff, as opposed to just MFI.
A lot of it's very similar to the microscopy software that Jim has up at the core facility, though. Like the region of interest feature that they use on the Nikon software is pretty much works the same way as creating that binary. You define a threshold, and then you apply the binary onto the image. So if you've done one, then the other follows pretty easily.
That's very good to hear, David. I'll give Jim your shout out. That's really kind of you. He's traveling today, otherwise I'm sure he'd be here. But maybe there's an opportunity there, Amy. We can think about letting more folks know who are in the transition between technologies, how they can come down and see if the Amnis will help them too. So.
Yeah, I think the power that David spoke to is its numbers, in a shorter period of time. If you want really pretty images, you definitely want to go to Jim. But the throughput is a little faster, and the numbers our statistics is nice with the imaging flow for sure.
Any other questions from folks in the audience?
If not, feel free to reach out to me independently, and I'm sure David would be happy to answer any questions. Thank you all for coming.
The link to the core in the chat, and also the link to our core seminars. Do please feel free to check those out and let us know. Amy's contact information is in there. And the next couple of days, if not within the next week, we'll have the seminar up for you to review and share with your colleagues if they didn't get a chance to join us today. Our next seminar will be May 10th from 2 to 3, same time. And we're going to be highlighting the mass spec core facility that day. So we look forward to seeing you there. And if anything comes up between now and then, please feel free to reach out. Thank you all for joining.
Thanks, Andrew. Thank you, David.
Thanks.
Great job.
Thank you.